photosynthesis, and for further information you go to this photosynthesis Wikipedia article, separate article on it. So it's going further. So photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy that can later be released to fuel the organism's metabolic activities via cellular respiration. This chemical energy is stored in carbohydrate molecules such as sugars, which are synthesized from carbon dioxide and water. In most cases, oxygen is also released as a waste product. Most plants and algae are a large group of photosynthetic eukaryotic organisms. And cyanobacteria perform photosynthesis, which is largely responsible for producing and maintaining the oxygen content of the Earth's atmosphere and supplies most of the energy necessary for life on Earth. Yeah, so basically CO2 plus water, you get energy from that as a form of carbohydrates, sugars, and oxygen is also released as a waste product in most cases, and oxygen is needed for breathing. And needed for pretty much for life on Earth, and cellular respiration, or it's energy production. So MES note: an atmosphere from the so this is maintaining the oxygen content of the Earth's atmosphere. So an atmosphere from the Greek words right here meaning atmos, which means vapor, and this one's spharia. Yeah, or sphera, meaning ball or sphere, is a layer or a set of layers of gases surrounding a planet or other material body that is held in place by the gravity of that body. And note the words, so atmosphere also could be broken down as atom sphere. So the surroundings of the atom or the Earth. So visualization of composition by volume of Earth's atmosphere. Water vapor is not included as it is highly variable. Each tiny cube, such as the one representing krypton right here, all this super tiny one all the way across there, has 1/1,000,000th of the volume of the entire block. So each block has 1/1,000,000th of the entire one, which is absolutely fascinating. So basically you have this one here, nitrogen is N2 makes up 78% of the of the atmosphere. Then you have oxygen in this white one right here that makes up 20.9%. Then you have argon in this green layer right here, which is 0.934%. CO2 is this right here, so it's 0.037%, so very, 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 very tiny. And then you have neon right here, even smaller, so this is 0.0018. And this ppm is parts per million. So basically all these little parts per million. So this is 1/1,000,000th of of a block. Each block is 1/1,000,000th. So this is how many of these inside. And then you have what else is helium is even tinier, 5 parts per million. CH4 is 2 parts per million. That's methane. And then krypton is 1 parts per million. And then N2O, nitrous oxide is 0.5 parts per million, and then H2 hydrogen is 0.5 as well parts per million, so 0.00005%. Absolutely fascinating. Let's continue further. All right, so photosynthesis has 4 stages: light absorption, electron transport, ATP synthesis, and carbon fixation. Yeah, so the first stage, light absorption, is the initial step of photosynthesis, whereby light energy is absorbed by chlorophyll pigments attached to proteins in the thylakoid membranes. And so thylakoid membranes are membrane-bound compartments inside chloroplasts and cyanobacteria. And here I just added just a brief note on cyanobacteria. Yeah, I think that's how I pronounce cyanobacteria. So it's a type of bacteria that obtain energy via photosynthesis, and we'll go over bacteria later in this video. They are the site of the light-dependent reactions of photosynthesis, and thylakoids consist of a thylakoid membrane surrounding a thylakoid lumen, which is lumen is inside a membrane or inside a tube or a opening or vessel, etc. Chloroplast thylakoids frequently form stacks of discs referred to as grana, singular is granum, and grana are connected by intergranal or stromal thylakoids, which join which join granum stacks together as a single functional compartment. And the thylakoid membrane is a site of light-dependent reactions, photosynthesis, with the photosynthetic pigments embedded directly in the membrane. It is an alternating pattern of dark and light bands measuring each of 1 nanometer. Again, that's 1/1000th of a meter. Fascinating stuff. So here is a diagram of it. So thylakoids, dark green, and inside a chloroplast. Yeah, so here's the thylakoid right here. So these dark greens pointing at these ones right here inside the chloroplast. And remember, thylakoids is the site of light-dependent reactions of photosynthesis. Yeah, so it consists of a thylakoid membrane inside the thylakoid lumen. So lumen's inside, and then the membrane is, yeah, the super dark green is the membrane there, and then inside the lumen. And this is the inner membrane is for the chloroplast, and this is the outer membrane inside a chloroplast. And then the granum are the stacks of it. So chloroplast thylakoids frequently form stacks of discs referred to as grana, or in singular is granum. And granum are connected by intergranal or stromal thylakoids, which join granum stacks together as a single functional compartment. So you have a bunch of granum stacked together. And this stroma is just the part of a tissue or organ with a structure or connective role. Yeah, so these are the connected by intergranal or stromal thylakoids. So it's all connected by here. So so this one inside here, so acting as a connective tissue, is part of the tissue organ with a structural connective role. So this one could be considered as a stromal thylakoid. So this is intergranal or stromal. Yeah, or stromal. I think that's how you pronounce it. I just Google search that, the pronunciation. And then there it is. It's forming these stacks called granum. Granum is singular, and then for one of them, and then grana for multiple. So there's 3 grana or 4 here. And these are the connective or stromal thylakoids. So stroma is the part of the tissue or organ with a structural connective role, and granum are stacks of thylakoids. Going further, here's another illustration. Here's the diagram. So thylakoid structure. So if you this entire thing is this one in red is a granum, and this one is also called the laminelle or laminella. Or actually, it's pronounced lamella. Yes, the search out. And this is the inside these thylakoids. You have a thylakoid lumen, and there's the membrane around it. That's a super dark green one over there. Let's continue further. Now here's a electron microscope image. So let's make this a bit smaller. So we could fit this in. Just make it even smaller. Yeah, so this is fascinating. So let's make it a bit bigger. Even bigger. Let's okay, make it bigger. Right here. Okay. So now we have here. So scanning electron transmission. Scanning transmission electron microscope or STEM imaging of thylakoid membranes at 10 nanometer thick. STEM tomographic slice from a lettuce chloroplast. So that's a very, very, very small. So the thickness of this is, if you have a 3D diagram like this, whoops. So 3D diagram like that. If this is 3D, this is going to be from here to here. It's going to be 10 nm, which again, it's a billionth of a meter. So let's erase this. So that's pretty fascinating stuff. And here I quickly just changed the size again, just made all these smaller to fit this in this page right here. And so this is 10 nm or 10 nanometer thick stem tomographic slice from a lettuce chloroplast. So this is from lettuce, pretty interesting. Grana stacks are interconnected by unstacked stromal thylakoids called stroma lamellae. And scale bar is 200 nm nanometers. This scale bar right here, 200 nm. Very, 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 very tiny. So there's the grana stacked up like that, and there's a stroma laminelle, so the extended part of this thylakoid structure there. So yes, and look at how these structures are all like that. There's the grana stacks, and then there's the stroma lamella or lamellae. Yeah, or I think this is pronounced lamellae. So yes. So the AE might be sounding like an I, so I have to search that up. Anyways, it's fascinating stuff. That's an electron a microscope image of it. So yes, it actually looks pretty decently like the animation there, or the diagram. And going further, Yeah, going further, we're going to look at absorption right here. Because remember, the thylakoid is a site of light-sensitive chemical reaction or light-dependent reactions of photosynthesis. And right here, the first stage is the initial step of photosynthesis is light is absorbed by chlorophyll pigments attached to proteins in the thylakoid membrane. So on the membranes, it's going to be absorbed. So light's going to be absorbed on these membranes in the chlorophyll. Yeah, chlorophyll pigments, the green color pigments attached to the proteins in the thylakoid membrane. So attached right on them, and basically now let's look at what light absorption means. So absorption or light absorption of electromagnetic radiation, which is just a general light wavelengths, is how matter, typically electrons bound in atoms, take up a photon's energy. Yeah, so the electrons, typically contain that energy. It's very interesting. So the photon's energy, and so it transforms electromagnetic energy into internal energy of the absorber. For example, thermal energy. So in other words, so you have an external radiation, considered as a vibration in the ether in MES science, and then it shakes the electrons or spins them, and then the energy is directed into these electrons locally. So you get external energy into local. A notable effect is attenuation, or the gradual reduction of the intensity of light waves as they propagate through a medium. Yeah, so as it goes through a medium like water, etc., it gradually gets weaker and weaker. So here is an overview. Here's a diagram right here. It's an overview of electromagnetic radiation absorption. This example discusses the general principle using visible light as a specific example. A white light source emitting light of multiple wavelengths is focused on a sample, and the pairs of complementary complementary colors, which are opposite colors and cancel when combined or mixed, and have the highest relative contrast to each other, such as black and white. They're complementary colors, are indicated by the yellow dots right here. So we'll go over here. So you have a beam source right here, emission of these lights, wavelengths right here. So you have white light, a bunch of different wavelengths, and these dotted lines indicate the complementary colors there. And then incident radiation. So basically, these are the photons that strike this sample, and then this gets absorbed inside. So we'll continue reading here. So right here, upon striking the sample, photons that match the energy gap of the molecules present, so green light in this example, are absorbed, because hence the pigments, you need the specific energy matching, or wavelength matching, correlating to the size of the pigments and so on. Exciting the molecule. So if you have green light hitting these molecules, they're small enough to interact with it, with this vibration or light photons. So the energy of this molecule gets increased, and then so notice how it's just increased. There's that spinning illustration of it, so you can view it as the electron spinning or the molecules all speeding up or vibrating and getting excited. So there's these green ones getting struck by this and excited. So the light in this case, green light in this example, that excites the molecules. Other photons are transmitted, other photons are transmitted unaffected, and if the radiation is in a visible range, 400 to 700 nanometers, the transmitted light appears as the complementary color. Here is in red. So green, red are opposites. When green is absorbed, reddish color is observed. So the contrasted complementary color, the opposite of the green, is red. And by recording the attenuation or loss of intensity of light for various wavelengths, an absorption spectrum can be obtained. So you have basically light of a bunch of wavelengths coming in, and then the green gets absorbed, so what gets shown is the red, the complementary color of it. And there's absorption, there's transmission, there's detection, so you detect the color that's being shown. You detect the color being shown, then you can see which one was absorbed, and you can also see this is green, it's reflecting back. And then what you're left with is here. So if these are complementary colors, so we have the red and green right here, and then these other ones are complementary over there, all you're left with now is just the two. And the red one, so you have the red, but the green one's gone. So the gone, that gets absorbed over here. So that green light gets absorbed into it, does not get transmitted, or the wave of the ether does not get transmitted. And then here's a spectrum right here. So if you have a number of photons that go through, this transmission spectra, and then the absorption is this green one. And then that's a transmission spectra, or a spectrum. It's showing you all the wavelengths, and then all the ones across here that gets detected here. And then all the ones that get absorbed, you can calculate it by the ones detected, and you can see that's over here. That's going to be the green one. It's going to be opposite. You could also just see this one and measure the reflected ones across. So yeah, it's very, very fascinating stuff. So that's just a quick general overview of absorption, and that needs a video on itself in later videos. To go in more detail on it because it's very fascinating stuff. Alright, so now let's get back into the script. So the absorbed light energy, and then just scroll back here again. So remember, the absorbed light energy, or the light absorption, is the first stage of photosynthesis, and light absorption is involved where light energy is absorbed by chlorophyll pigments attached to proteins in the thylakoid membranes, or thylakoid. Yeah, thylakoid membranes. So in other words, right on these membranes on the top here, on the outer surfaces of them inside the chloroplast or cyanobacteria. Yeah, so on the membranes there's photosynthetic pigments embedded directly in the membrane. So the thylakoid membrane is the site of the light-dependent reactions of photosynthesis, with the photosynthetic pigments embedded directly in it, and where it first absorbs the light. So let's go over here now. So the absorbed light energy is used to remove electrons from a donor, usually water, to a primary electron acceptor called a quinone, and this is an oxidized derivative of aromatic compounds. And we actually know this already. If we scroll back here from the beginning, we already mentioned quinones earlier when we talked about FAD. Yeah, which in the citric acid cycle, the total yield from the cycle was 1 glucose or 2 pyruvates, 6 NADH, 2 FADH2, and 2 ATP molecules. And then the FAD is, or flavin adenine dinucleotide, is a redox active coenzyme associated with various proteins, which is involved in several enzymatic reactions and metabolism. And FAD can exist in 4 redox states, which are the flavin N5 oxide, and then there's the quinone. So let's bold this right here. And then semiquinone, and then the hydroquinone. And also note that FAD is converted between these states by accepting or donating electrons, and FAD in its fully oxidized form, or quinone form. Yeah, so it accepts 2 electrons and 2 protons to become FADH2, or the hydroquinone form. And now what I've done is just copy and pasted more info, including just a comparison chart. And just the reason for this is, let's go back here again, going back over to this part right here. Yeah, this is the one we just read here. So the quinone is oxidized derivative of aromatic compounds. All right, and now we are back here. So basically, this quinone, so FAD in its quinone form, is an oxidized derivative of this FAD, and FAD is an aromatic. It has an aromatic ring system, or it's a aromatic molecule or compound. So continue reading further. So the semiquinone FADH, and here I just noticed it actually has a dot there. So this FADH dot. Note the dot indicating it is a free radical, and this can be formed by either reduction of FAD or oxidation of FADH. So to oxidize this, you got to take electrons out. To reduce this one, you got to add electrons to it by accepting or donating 1 electron and 1 proton, respectively. So in the first case here, to oxidize this, you accept the electron and proton, or just the electron you're accepting. It's going to be, to reduce this one. So to reduce this one, you accept the electron. And then to oxidize this one, you have to donate an electron. From this. Yeah, this one is a reduction of this requires accepting. And then oxidization of FADH2 requires donating. And also this free radical, just to recall what free radical was. So if I go to radical, if I just do a search for radical, let's go here. All right, so there it is. We had it actually earlier. Actually, no, it's later in this video. That actually already covered it. So this, remember the radical right here. A radical is an atom, molecule, or ion that has at least 1 unpaired valence electron, and sometimes it's called a free radical. So you have an unpaired dot like that. So there's an unpaired electron on this, and this makes it more reactive to form chemical compounds, or react with other species, other chemical compounds and molecules, and so on. Continue further in this, just to go more info on this and illustrate how they all correlate with this quinone topic. So some proteins, however, generate and maintain a superoxidized form, the flavin N5 oxide. FAD is an aromatic ring system, so hence a quinone is an oxidized derivative of an aromatic compound, so FAD. So you oxidize FAD by taking electrons from it, then you get a derivative of it. And that derivative, you're deriving a compound from this, and that's the quinone. So you have the quinone, semiquinone, the hydroquinone, and this superoxidized one, just call it oxide there. So going further, so FAD is an aromatic ring system, whereas FADH is not. And if we recall aromatic, it's just it has resonance and it's super strong. So aromatic. Let's go here. Aromaticity is a property of cyclic, ring-shaped, planar, flat structures with pi bonds and resonance, those containing delocalized electrons that give increased stability compared to other geometric or connective arrangements with the same set of atoms. Aromatic rings are very stable and do not break apart easily. Organic compounds that are not aromatic classified as aliphatic. They might be cyclic, but only aromatic rings have enhanced stability. So you have enhanced right here, has delocalized electrons, and it's super, super strong. You have it in different forms. You, if it gets shifted over and you have different setup on the electrons, it will just exist in the other setup, and it keeps, so you can find it alternating or find these similar patterns right here in a hybrid right here. Yeah, so two different resonance of benzene top combined to produce an average structure. So it's fascinating stuff. So basically super strong and has resonance with the delocalized electrons that don't have to stick to one atom. Alright, so just teleport back here. Yeah, so FAD is an aromatic ring system, whereas FADH2 is not. This means that FADH2 is significantly higher in energy without the stabilization in through resonance. And remember, this is the oxidized version, so it has... Actually, no, it's not. This one's not oxidized. This is the reduced version. So this one has electrons, a bunch of them inside, so it has high energy, but it's not that stable. So without the stabilization through resonance that the aromatic structure provides. And FADH2 is an energy-carrying molecule because once it oxidized, once it removes its electrons, it regains aromaticity and releases the energy represented by this stabilization. So here is a comparison, different redox states of FAD. So you have here this is the superoxide version. So let's look at its fully oxidized form as the quinone form. That's actually over here. This one's a superoxidized one. So superoxidized as a form right here, and it has aromaticity. Notice how the rings, you could switch up and have different setups over here. Yeah, but in this diagram it just shows this N and this one without it. NH, this one without the H there. But this one right here, you have the ring structure like this. This one, it can go here, it can go on the top, and so on. But anyways. Yeah, I can't validate exactly this diagram, but yeah, you get the idea. So it could have it has different types right here, with an H or without. So in this case, if it loses an H, it goes there. But then now, so if you have this quinone, this is fully oxidized. So now it's fully oxidized. So if it accepts two electrons, it accepts an electron and hydrogen, it gets to this semiquinone form. So it's reduced. And then if it accepts another one right here, and then notice the dot right there is the dot for the unbalanced electron pair. Hence, it is a radical. So there's a free radical as well on this one. So different forms of a semiquinone. And then going down again here, if you take an electron and an hydrogen to reduce this, one, you get this hydroquinone right here. So you have this structure, and it has variations right here. This is with, and then if you remove an H, it gets to this. If you add an H, it gets over here. And then going backwards here, so if you oxidize this by removing an electron, you get the semiquinone. Oxidize again, fully oxidize, you get this quinone. But if you go superoxidize it, as is stated right here, some proteins, however, generate and maintain a superoxidized form, the flavin N5 oxide. So then this one takes another one out of here. Yeah, this one right here, this is actually 2 electrons and 2 protons. If you take those out, you get this over here, the superoxidized form. And then if you add them here, I think, yeah, this takes it from the water there. So yeah, it takes it from water to get this superoxidized form. But then if you were to go reduce this, you would add 2 electrons, and then those form water. You add 2 electrons, and you also get water released here, and you get it this form, and so on. So it's fascinating stuff. So let's go back over here. Quinone. Yeah, so right here, this is the water one. So absorbed light energy is used to remove electrons from a donor, water, to a primary electron acceptor, a quinone, and this is the oxidized derivative of aromatic compounds designated as Q. Alright, so now in the second stage of photosynthesis, electrons move from the quinone primary electron acceptor through a series of electron carriers until they reach a final electron acceptor, which is usually the oxidized form of NADP+, which is reduced to NADPH, a process that takes place in a protein complex called photosystem I, or PSI. Yeah, so this oxidized form here, it's reduced by gaining electrons to NADPH. We'll get to that in a bit, but let's just scroll up into these 4 stages. So photosynthesis has 4 stages. Light absorption, electron transport, so this is the electron transport stage, and then it has ATP synthesis, and then carbon fixation. So we've covered light absorption, now the electron transport. Alright, continue further. So the transport of electrons is coupled to the movement of protons or hydrogen from the stroma to the thylakoid membrane, which forms a pH gradient across the membrane as hydrogen becomes more concentrated in the lumen than in the stroma. And this is analogous to the proton-motive force generated across the inner mitochondrial membrane in aerobic respiration. And note the similarities. Basically, movement of electrons, and then you create a gradient where you'll have protons being forced to move elsewhere and create a force. Yeah, so the transport of electrons is coupled to the movement of protons or hydrogen from the stroma to the thylakoid membrane. So the stroma is just a part of a tissue or organ with a structural or connective role. And here, actually, I just forgot to make a better clarification, so I just double check this. Yeah, so this stroma is this fluid as well. So stroma is a part of a tissue organ with a structural or connective role. So that's this inside over there. And specifically in botany refers to the colorless fluid surrounding the grana within the chloroplast. And botany is just the study of plants, as I alluded to earlier, or the plant biology. So yes, that's this over there, the stroma. And yeah, so what happens here is going back here and read this again. Yeah, so the transport of electrons is coupled to the movement of protons from the stroma to the thylakoid membrane, which forms a pH gradient across the membrane as hydrogen becomes more concentrated in the lumen than in the stroma. So it moves from, so just bold this, so from the stroma. So you'll have electrons transport, and then it's coupled with the movement of protons inside from the stroma, inside the lumen. Yeah, inside the thylakoid, or specifically the thylakoid membrane. So it just basically goes to the membrane and so on. But then what this will create is a gradient where the protons are more condensed in this lumen area, so inside and in the membrane and so on, than the outside. So then this is going to get a force that wants to push outwards. Alright, so let's look at that again. So the transport of electrons is coupled to the movement of protons or hydrogen from the stroma to the thylakoid membrane, which forms a pH gradient across the membrane as hydrogen becomes more concentrated in the lumen than the stroma. Lumen is the inside of the thylakoid, or any vessel, and so on. This is analogous to the proton-motive force generated across the inner mitochondrial membrane in aerobic respiration. Let's go further. So this is the NADP+. So this is basically in the second stage, electrons move from the quinone primary electron acceptor through a series of electron carriers until they reach a final electron acceptor, which is usually the oxidized form of NAD+, which is reduced to NADPH. And then in this process takes place in this PSI, or this protein complex. So this NADPH+, so nicotinamide adenine dinucleotide phosphate, abbreviated NADPH+, is a cofactor used in anabolic reactions. And again, cofactor is assists. It's required in enzymes activity or activity of behaving like a catalyst or activity as a catalyst. So it is used in anabolic reactions, which is the building up of molecules such as the Calvin cycle and lipid and nucleic acid synthesis. And I'll get to the Calvin cycle in a bit. So which require NADPH as a reducing agent. So requires NADPH as a reducing agent, where NADPH is a reduced form of NADP plus. So this is oxidized. This is lost electrons, and then this is it gains electrons to reduce to NADPH, and then this NADPH. Yeah, this NADPH acts as a reducing agent, and I'll just put this here, further info from Wikipedia. Just copy this here. Actually, I already have this here, yeah. I just had a little brain fog. I'm just going to delete this. Okay, so we already have that there. So NADPH is the reduced form of NADP+, and NADP+ differs from NAD+ by the presence of an additional phosphate group. Yeah, so this is the reduced form, so it gained electrons. So NADP plus gains electrons, and NADB plus, this differs from NAD plus by the presence of an additional phosphate. So yes, very, very similar here. This has a phosphate, and then otherwise you can have NAD and nicotinamide, adenine dinucleotide. Yes, and then or if you had the phosphate, you get this. You get this right here, NADP plus, and then if you remove that, you get this NAD plus. Yeah, so it acts as a reducing agent. So what this means is it reduces others. So if it's already reduced and it has electrons, so then the electrons would give off to other molecules, and then when it gives off to others, yeah, so when you give this E minus or this negative charge to another another molecule, then this becomes reduced, so its oxidation state goes down. So its O goes down, and then this gets oxidized. So this becomes oxidized to go there because this has a reduced form of NADP plus, so it already has electrons. So it gives off to it. So it reduces the molecule. So this is a reducing agent, while this oxidizes. So it oxidizes itself, but by reducing others. So yes, it is used by all forms of cellular life. So fascinating stuff. We continue further. So during the third stage of photosynthesis, the movement of protons down their concentration gradients from the thylakoid lumen to the stroman through the ATP synthase is coupled to the synthesis of ATP by that same ATP synthase. Again, that's an enzyme that speeds up the synthesis of ATP, and it's required for most circumstances. Yeah, most or a lot or common. Continue. So this is the third stage of photosynthesis. Let's look at that again. So yes, if you see, this is how I learn. I keep going back and forth until it gets stuck into my brain. So that's ATP synthesis. So we're at here. The next one's carbon fixation. So we went from light absorption, electron transport, and ATP synthesis, and carbon fixation is the next one after this one right here. Yeah, so the movement of protons down their concentration gradient from the lumen of the thylakoid to the stroma, or that fluid around the grana but inside the chloroplast, through the ATP synthase is coupled to the synthesis of ATP by that same ATP synthase. And right here, to illustrate further on this concentration gradient, it has to do with molecular diffusion, or often simply called diffusion. It is the thermal motion of all liquid or gas particles at temperatures above absolute zero. So what happens is molecules tend to move from high to low concentrations. So high to low, this basically means there's a concentration gradient, and so molecules tend to move from the high concentration to the low one. So here's an example right here. So if you have here, this is to set up these molecules. So you have one first. We'll pause this. Actually, we'll go back. Yeah, we'll pause it actually. Okay, so we have it like this. So in this case here, so here's three types of gradients. We'll make it smaller. So diffusion from a microscopic and macroscopic view. So let's just play this. Yeah, so initially we have, before we started, so we have it like this. So we have a barrier here. So this is high concentration. This one is the same concentration everywhere, so it doesn't really change anything. And this one here is a barrier as well. And then once we remove it, this is a high concentration of this blue color. And once you remove it, what ends up happening is this wants to blend over to this side, and these balls want to move over to this side. But this one doesn't really matter because it's pretty much same concentration. Yeah, so I'll just put this away. How do I cover that sign? Okay. Yeah, so cover that up, and notice how this moves to the right. So that's essentially what that happens at proton-motive force. You have a concentration gradient of protons, and then they want to separate away from each other. And it keeps going, especially since like charges repel. Alright, so continuing further. So the NADPH and ATPs generated by the light-dependent reactions in the second and third stages respectively provide the energy and electrons to drive the synthesis of glucose by fixing atmospheric carbon dioxide into existing organic carbon compounds, such as ribulose biphosphate or RUBP, in a sequence of light-independent or dark reactions called the Calvin cycle. Alright, so remember the second stage was the electron transport, and involves this being generated. And if we scroll back up to that part, so we have here, so electrons are transported from the primary electron acceptor through a series of electron carriers until they reach a final electron acceptor, which is usually the oxidized form of NADP+, which is reduced to NADPH. So this is the reduced form once it accepts all the electrons. So in that electron transport stage, so now you have NADPH. And then the third step was the ATP synthesis. So the movement of protons, which is coupled from with the electron movement. And then movement of protons down their concentration gradient from the thylakoid lumen to the stroma through the ATP synthase is the enzyme is coupled to the synthesis of ATP by that same ATP synthase. So yeah, so you have the protons moving from the lumen to the stroma through. So here's a key word. So through the ATP synthase, and it's coupled to production of ATP. So you have ATP and you have NADPH. And these are generated by light-dependent reactions in the second and third stages, respectively. Provide the energy and electrons to drive the synthesis of glucose. Yeah, so now it's to drive the synthesis. Now you got to create the glucose now. So you have ATP, you have NADPH. So you have a bunch of electrons, and you have a bunch of energy molecules, or the energy currency, and now you want to actually produce glucose, which is energy. So the sugars, where you're going to eat. Okay, and also this is basically storing air. So this is the energy. So this is the electrons, and then you have energy molecule or currency, and now you want to produce synthesis of glucose for others to use that energy, or to store for later. Anyway, so you drive the synthesis of glucose by fixing atmospheric carbon dioxide into existing organic compounds such as ribulose biphosphate RUBP in a sequence of light-independent or dark reactions called the Calvin cycle. So basically, we're going to take this NADPH and ATPs, and it gives us the energy and electrons. The key one is electrons for NADPH, which is the final electron acceptor, and then to drive production of glucose. And it's also going to fix CO2, meaning you're going to store it inside existing carbon compounds, such as this right here. And these are light independent or dark. And here's an illustration of this Calvin cycle right here. Let's make this smaller. Yeah, so basically what we end up having is this. So here's the Calvin cycle. So just make it so that we can see. Yeah, so we have light going here. Light reactions involves H2O, and it releases oxygen as a waste product from plants, and then where animals and so on need it. They need it to breathe, so we need the oxygen for respiration or breathing and so on, energy. And then so basically what this light reaction produces ATP, NADPH, and then it also gets produced as NADP plus and ADP plus inorganic phosphate, or this is that phosphate in the solution. But this is after the Calvin cycle. So this fixes CO2, so it absorbs the waste CO2 in the atmosphere. Yeah, plus this ATP and ADPH. So hence why in the mainstream climate crisis scenario, where they're always complaining that there's too much CO2 and forests are being deforested and so on, so you want to build forests to grab CO2. Again, that's the mainstream narrative. So you have CO2, ATP plus NADPH, it's Calvin cycle, and it produces sugar. And this is the electrons here. This is the E minus. and this is energy. So there's E minus and energy, and then what you end up producing is diphosphate right here. I mean P plus. So this becomes oxidized. So this reduces. So this oxygen goes in here. So then what you're left with is NADP plus. So you take the electrons, and then this phosphate is also used in this to create to be used as energy. And in the reaction, what ends up happening is you remove a phosphate, and that phosphate becomes in solution. So basically uses up the energy and splits up this triphosphate into a diphosphate. And in the production is sugar or glucose. Just fascinating stuff. Yeah, so photosynthesis changes sunlight into chemical energy, splits water to liberate O2. So yes, this is the water there. When it splits it creates this over there. This is fascinating stuff. And then fixes CO2 into sugar. So the CO2 goes into the sugar. So now it's out of the atmosphere into the sugar, the compound, and you can eat it. And so on. And hence why you eat food. Yeah, the mainstream says population is too much and CO2 is growing or something like that. Anyways, continue further. And now let's discuss carbon fixation. So just note, carbon fixation or carbon assimilation is the process by which inorganic carbon, particularly in the form of carbon dioxide, is converted to organic compounds by living organisms. The compounds are then used to store energy and as structure for other biomolecules, and carbon is primarily fixed through photosynthesis, but some organisms use a process called chemosynthesis in the absence of sunlight. And let's just quickly discuss chemosynthesis. So chemosynthesis is the biological conversion of one or more carbon-containing molecules, usually carbon dioxide or methane, and nutrients into organic matter using the oxidation of inorganic compounds. Yeah, so the taking of electrons from these to increase their oxidation state. IG, hydrogen gas, hydrogen sulfide, which is H2S, or ferrous, containing iron ions as a source of energy rather than sunlight in photosynthesis. Yeah, so instead of getting energy from photons, you get energy from electrons using this oxidation. Yeah, oxidization of inorganic compounds such as hydrogen gas, hydrogen sulfide, and ferrous ions. It's fascinating stuff. The Calvin cycle, which is right here. We want to discuss this one. So the Calvin cycle, or also called light independent reactions, biosynthetic phase, dark reactions, or photosynthetic carbon reduction PCR cycle of photosynthesis are the chemical reactions that convert carbon dioxide and other components into glucose. So it's a part of the photosynthesis that's not dependent on light. So you have the light coming here. Here's the light reactions, and then you get this ADP, NADPH, and then it fixes the CO2. You get sugar, and then you get this diphosphate, and you get this this NADP, and then you also have this ADP, and then this is plus. So you take away the electrons and the hydrogen. Yes, you form sugar, and then you have this ATP triphosphate gets to a diphosphate and an inorganic phosphate, and then afterwards in the light reactions you have O2 is the waste. But basically the Calvin cycle is over here. So this is the non-light dependent, and not dependent on light. So independent from that, you get CO2. Once these are created, you get the sugar and so on, and you get these waste. Yeah, and these ways they go back into the light reactions over there. So it's fascinating stuff.